Professor Felipe Vadillo. He is a professor of the School of Medicine here in the university, and it's a great pleasure to have you here with us. And he's going to talk about vaginal microbiome and things like that. Thank you, Professor.
Thank you, Max. Max just told me that you have had a very nice meeting, and I'm hoping this is not going to disturb very much the quality of this nice meeting. I'm going to present you this area of interest in my group that is primarily interested in the topic of preterm birth. That's the field of the main field in my group, and we have been studying this problem, this health problem, since many years ago, and the part that I am going to present you this afternoon is related with an explanation for this disease. And we know this preterm birth is a very common condition around the world. And recently, the WHO launched an initiative regarding the relevance of the preterm birth around the world, and claiming that this is a huge problem. I hope I can convince you that this is really a huge problem for the health systems. And we know that preterm birth is defined this way. This is the most accepted definition of preterm birth. And it was proposed several years ago, but actualized by the WHO some years ago. And we define preterm birth as any birth before 37 weeks of gestation. So this is a big problem because all these reasons. It's really frequent. We have almost 15 million every year, every new cases of preterm birth around the world every year. So the meaning of this is that during this talk, we can expect that something like 150 newborns are born preterm around the world. And in Mexico, we can expect that in this hour, we will receive 20 new babies during this small time. So for the frequency, this is a big problem in every country. But it is also a big problem because it's a direct cause of death in the neonates, and is the main cause of neonatal death, and it accounts for the most important reason explaining deaths in boys less than 5 years old. So this is why this is relevant for the Millennium Goals. This is something that is related directly with one of the Millennium Goals that we are pursuing in the next years. And in addition to this being a direct cause of death, it is also related with many consequences in health in the future health of the surviving children, because it's associated with damage mainly to the central nervous system, and it is associated with cerebral palsy, for example. That is the main explanation for that. And the damage can range from very simple conditions such as loss of vision or poor perception of flavors to very deep damage to the central nervous system. So it is really costly. For us in Mexico, this is impossible to cover. this is the amount that the United States is spending in the attention of the babies derived from the preterm birth. And you can see that it's a really huge amount of dollars. We don't have that money in Mexico. So the meaning of that is that our boys, most of the boys that are born preterm, they are dying. We don't have enough beds around Mexico to cover all the numbers, all the babies born preterm. So the problem is a worldwide problem. You can see in this map, this is the most recent version of the rate of preterm around the world, and you can see that it's everywhere. The problem is everywhere, ranging from 5% to 15 to 20%, being the darkest color countries with the highest rate. Colombia, this is Mozambique, some areas in Asia that they have the highest rates of preterm birth. But you can see that this is not only a problem of poor countries, because we have just the same rate as the United States and very equivalent to the rates in Europe. So there is something around all these countries that is explaining these rates of preterm birth around the world. Maybe if you think in the African countries, you can see that they have the highest prevalences in the world, but and it's clearly related with poverty. But we have that problem. And the United States, they are experiencing a problem of high increase of the numbers in preterm birth rates. And they are going from numbers like ours. Our prevalence in Mexico is around 9%. And maybe some years ago, United States and Canada, they had the same numbers as us, but now they are going up, and they are now around 12%. So they have, in fact, more cases of preterm birth, Canada and United States, than Mexico. We are having just the same numbers as many years ago. So what's related with the explanation for this disease? And I want to speak about only this part of the problem. There are many other explanations that have been proposed during the years that maybe can explain preterm birth. But the most important association that we know is related with the genesis of preterm labor is infection during pregnancy. And you can see in this graphic that most of the are related with conditions in which we do not detect infection, but clearly from 30% is going in different places in different numbers, but it's going from 25 to 40% of the cases in which we can detect an infection. So infections during pregnancy are related with preterm labor, and that's something that in our hospital we deal with very frequently. And we know that infection in this cavity during pregnancy, this is the intrauterine environment. And we can explain that something happening in this area, in the vagina and cervix, infections, can ascend to the intrauterine environment. And one of the most common and accepted explanation for infection of the fetus or the structures in the intrauterine environment are related with invasion from bacteria from this area to the interior with, in contact with the tissues, in contact with the fetus, or the penetration of these microorganisms to the intraamniotic cavity. So this is the most accepted explanation, but we can explain also the presence of infection during pregnancy, because something that is traveling, some microorganisms traveling from by blood can reach the placenta and cross to the fetus. And also an infection that is affecting the peritoneum, for example, the microorganisms can migrate to the intrauterine cavity. So many explanations, and it's something that is strongly accepted, at least in the clinical side, that we have to deal with the control of infection during pregnancy, because these conditions, a broad group of infections, are strongly related with preterm labor. But for our problem, it's really frustrating this condition that if we treat pregnant women with antibiotics, even in a prophylactic way, we don't prevent preterm labor. This is a big problem. This is, I am showing you this meta-analysis that was published some years ago. And we have recent publications confirming this conclusion, that even using antibiotics, you can see this is a lot of women treated with antibiotics, 5,000 women treated with any kind of antibiotics, and the prevalence of birth was not modified. So something is wrong with this, because if we accept that infection is the cause of preterm birth, so the use of antibiotics must solve this problem, and it is not happening. So we cannot use antibiotics, so preventing infection, to prevent preterm birth. The meaning of this is that the explanation is more complex than a simple infection. We have tried every single way every single antibiotic, and maybe we're in front of a problem of the adequate antibiotic, because the problem is that most common antibiotics don't reach in adequate amounts this compartment. So maybe that's an explanation, but there are a lot. Very recently, US team published a new approach trying to deal with this, and just the same findings. So something more is happening. So that's why we are trying to modify at least the hypothesis for explanation of the role of the microorganisms or infection during pregnancy as an explanation for preterm labor. And we know that the microbiome is now an emerging area of study, and is really complicated. And maybe you know very well that this microbiome is a group of microorganisms that are harbored in different areas of our body, and they are kept very constant in every single seed and individual. And we know also that they are very specific of every single area in our body. So the microorganisms living in the mouth are completely different to those living in the skin, and completely different to those living in the gut or in the reproductive tissues. So they are really specific, and we are thinking that maybe our look to the explanation of the infection as a cause of preterm labor has been very simple for now, and maybe we have to involve the participation not of a single microorganism, but several of them, working as an ecological system. That is the way we are seeing the role of the microbiome, every single microbiome in our body. Let me show you some previous descriptions of the it's only this slide covering the previous findings on the composition of the microbiota, of the vaginal microbiota in normal women. These are results from normal, non-pregnant women. And you can see every single column is a different patient. And you can see that we have a relatively low number of different microorganisms in the vagina microbiota. So the first thing is that microorganisms composing the vaginal microbiota are very few compared with those living in the gut, for example. In the gut, you can have a list of thousands of different microorganisms. In the vaginal microbiota, we have no more than 30 different microorganisms. And most of them are a group of microorganisms known as lactobacilli. And this group of microorganisms are dominant in this microbiota. You can see from this description that the composition in different women is different. Even from these few microorganisms, the composition is different. And you can see some clusters of distribution. And this is why they separated in these 5 groups. But some of those that we consider normal for the vaginal microbiota, such as those I just mentioned, they cannot be so abundant in some women. Look at this group of women. They don't have the same amount. This red color, or almost red color, is indicating the abundance of these microorganisms. So some of these women, they have large numbers of crispatus and other lactobacillus like inners. Other woman doesn't have that. And so there is a big distribution. Yes?
What about lactobacillus?
That's something that is a generic name of a group of different lactobacilli. Yeah. So all of them are acidophilus. Okay. That's a generic name for all this. That's the constant description for this lactobacillus. And we believe that these lactobacilli are participating in the secretion of acidic compounds, lactic acid, for example, that confers a very specific pH to the vaginal secretion that is enough to keep other microorganisms growing in that area, for example. So, and some others, you can see that almost the lactobacilli are the most important in every woman. But then we have this group of women here in which we found a lot of different microorganisms that they are not very abundant in the normal microbiota in this area, but they are really prevalent in some women, contrasting with the low amount of the lactobacilli. And all of them, as I told you, they are normal women. So we have versions. And this is important because every population may have a very specific composition of this bacteria. So we are trying to solve some of the pending issues in the study and the meaning of the microbiome studies. And so in this moment, we are dealing with first the description of the local composition of these microorganisms, because we don't have that information for Mexico, and we want to compare the composition of the microbiome with other populations. But we are especially interested, and I want to show you some preliminary results we have in this area. The bacteria, the microorganisms, are interacting with the immune system. And we don't know how this is happening. We don't know if the bacteria is affecting the immune system. We know that that's happening, in fact, but we don't know how the immune system modifies the local composition of the microorganisms. The most probable condition at the end is that we are going to see a mix of these both effects, and the resulting composition of the microbiota will be related with the immune system condition in these specific tissues. So we are exploring that, especially in order to explain the role of this microbiome with the preterm labor. And we look into the consequences of these interactions. So first, I want to show you some of the information we have on the composition of the bacterial communities in Mexican women, how they change during pregnancy, and at the end, I want to show you the consequences of these changes. Only to be very specific with the approach we are having now, we are exploring this area in the reproductive system of women. So we are exploring this area. We cannot explore during pregnancy other areas of the intrauterine compartment because of ethical restrictions. And so we believe that exploring this area, the vaginal microbiota and maybe some of the cervical microbiota, can be a good approach to what's happening in the intrauterine environment. In the School of Medicine at UNAM has a specific area at the National Institute of Genomic Medicine. This is located in the third floor of this building, and all this area is the embassy of the School of Medicine at the National Institute of Genomic Medicine, and my group is working in that area, and we are part of a microbiome consortium inside of the institute. And we are putting together all the resources we have, both the lab and the hospital's resources. And we have been working with the Secretaría de Salud of the local government in Mexico City. We have access to different hospitals of this network. There's CEDESA. They have at least 8 different hospitals around Mexico City, and they cover something like 50,000 pregnancies every year. And we have access to at least 3 of these hospitals. Our main clinical area is located in the Enrique Cabrera Hospital, that is a general hospital, and we are seeing women directly by our team, directly in this hospital. So we have also a specific area inside of the hospital in which we have our team working in direct contact with women. And for the first year, we have been working with the University of Michigan, and we had a joint project on the effects of air pollution on preterm birth and other consequences in pregnancy. And most of the money for this projects has been obtained through a grant from the NIH. So we are studying women living in Mexico City, different areas of Mexico City, and we are taking samples from them to study the microbiome composition that I am going to show you. So we have all the pipeline for collection, processing, and analysis of the microbiome. That is why we talk about the Microbiome Consortium at the National Institute of Genomic Medicine. And we are following women, pregnant women, starting very early during pregnancy. And we are following them every 4 weeks during gestation. So they are visiting us, our clinic, every 4 weeks, and we are following them for everything possible. So we are mapping these women for every condition. Of course, we are covering the normal OBGYN follow-up, but we have also following the fetal health because we have the specialists for the maternal fetal medicine included in the follow-up. So we are taking a lot of information from these women, and we completed the part of this follow-up when we finished with the first 1,000 women. So we have information and samples, biological samples, in a very large biobank and a really rich database that includes all this information. Complete medical history, the results from the laboratory tests, the maternal fetal medicine results, clinical nutritionist information, and we have even, this is something that is really important for us because we have a complete mapping of the diet of these women, and so we have a lot of information, and we are following this kind of women. They are in a very specific group. They are classified as normal women. We have also pregnancies that we are in a specialized center at the National Institute of Perinatology, but we are dealing only with the normal pregnancies. So from the beginning to the end. So I am going to show you the result we have in this follow-up of the cervicovaginal samples in relation with the microbiome. So I am going to separate some of these women that were classified as normal pregnancy and those coming for women that eventually developed preterm labor. And these normal pregnancies are delivering from 38 to 40 weeks of gestation. So spontaneously they have labor, they have single products pregnancies, and they don't have any other complications. So that's why we are talking about normal pregnancy. Preterm labor women that develop preterm labor, deliver between 32 and 36 weeks of gestation, and they don't have any other complication except the presence of preterm labor. Our pipeline for analysis of the microbiome is a standard methodology. We are extracting bacterial DNA with the devoted technique that is for bacterial DNA extraction, and we are amplifying both regions. We need this to cover, to deeply cover the sequencing of the and identification of the bacteria. That's why we are using B3 and B4 regions of the 16S RNA from the bacteria. So we are amplifying these two regions, and we're sequencing those regions using the MySeq classical equipment, and we are reading we are identifying the bacteria using simultaneously two different protocols, the classical KIME database and processing, and a correction that is a variant of the KIME process that is more stringent for the identification of the specific microorganisms. So I am going to show you some of the results we have. And in this graph, you can see that basically we are describing the results with diversity and richness. And if you compare the diversity of the preterm with term, you can see that at least in this part of pregnancy, we have a higher diversity in those microbiota of women developing preterm labor. And the richness of those bacteria is going down. These two are the combinations we can define as the characteristics of the microbiota in preterm labor. But I'm going to show you more. This is a specific composition. You can see here the different microorganisms, the major microorganisms that we are identifying. And during gestation, you can see that at the beginning, you can see some extra microorganisms in addition to lactobacillus species. Most of the microbiota is composed by the lactobacillus species. They dominate from the beginning, but going ahead during pregnancy, you can see that at the end of the pregnancy, the normal pregnancy, practically all of them are lactobacillus. Other species are disappearing from the normal microbiota, so this is a big change in the vaginal microbiota of normal pregnant women. And most of them are these two species, Crispatus and Iners. This is the genetic sum of those lactobacillus species. So what's happening during normal pregnancy in this group of women? They are going from a diversity dominated by the lactobacillus to almost the unique presence of lactobacillus species at the end of gestation. And what's happening with the women developing preterm labor, you can see that, as we suspected, there is a completely different panorama of composition. From the beginning of pregnancy, you can see that many different species are present in these women, and this is not changing the same way as the normal women to the end of gestation. And different amounts of these microorganisms in which we identify dominant species as G. vaginalis and Schuttler-Wartia, they are overexpressed in those microbiota from women developing eventually in this end of gestation, preterm labor. So that's why I mentioned that the richness is decreasing, because you can see that lactobacillus are less rich and the diversity is increasing. This is a combination that is associated with the microbiota of women that eventually developed preterm labor. This is summarized in this conclusion. So we can say that compared with non-pregnant women, that we don't have those women analyzed in our series, but we can use the reference from published information. We can say that during pregnancy, there is normally a decrease in diversity and increase in richness of lactobacilli. And that this vaginal microbiome is different from women going to normal term of gestation compared with those developing preterm labor. But we are interested in the consequences of these changes. And we believe that the change in composition in this area is affecting all tissues in this microenvironment. And we know that the hypothesis of relationship between infection and preterm labor is related with the presence of some inflammatory reaction in this area that we know as the choriodecidual area. This is the frontier between the mother and the fetus, and most of the evidence that my group and other groups around the world have provided is that that something is happening in this area, this choriodecidual area, and maybe this is the area reflecting the changes in the composition of the microbiome I just told you. And we believe that inflammation is a link between the modifications in the composition of the microbiota and the future of pregnancy. So we are exploring this relationship between presence of different microorganisms and preterm birth. And this is only a very simple association between the presence of different microorganisms, and some of them, these three in green, they are protecting, they are related with term labor, normal term labor. And all of this, that includes most of the species that we are identifying in the cases that I just showed, they are related in red with a risk for preterm birth. So most of these species, in clear relationship with the composition of the microbiota, these are associated with a high risk for development of preterm birth, and these three are related with the normal delivery and normal labor. But we want to see if the contact between these microorganisms and the intrauterine tissues results in some reaction of the immune system. And we are taking samples of these secretions in this area, the cervicovaginal area, and we are taking swabs of those areas and looking for the expression of different cytokines related with the inflammatory reaction. And we are using a multiplex system to detect all these 19 different cytokines in these fluids and relating the concentrations with the presence of normal labor and preterm labor. And this is another simple association between the mean values of the cytokine concentrations in these two groups. And I am showing you the specific microorganisms in this group of women who developed preterm labor, and those same microorganisms with term labor. This is reflecting only the association between the presence of increasing amounts of these microorganisms and increasing amount of all these cytokines. These 19 cytokines are reflecting both the pro-inflammatory and the anti-inflammatory cytokines. And you can see very clearly that in those cases with preterm labor, those women are secreting more cytokines, inflammatory cytokines, as in this broad approach to the problem. But it's very clear that women, normal women, not related with higher amounts of high amounts of the cytokines. And here you can see that when we compare, this is the microbiota composition term that and preterm that you saw a few moments ago. And this is the inflammatory cytokines group. Every color represents a different cytokine. I didn't put the specific names of those cytokines, but I put all of them together as pro-inflammatory or inflammatory cytokines and anti-inflammatory cytokines. So the change in the composition of the microbiota in women developing preterm labor is clearly correlated with an increase in the amount, the sum of those cytokines that are related with the pro-inflammatory response. It's really clear. And you can see here that we have almost constant amount of the cytokines in the normal women, that they are not showing these changes in the composition of the bacteria. And on the other side, you can see here that the anti-inflammatory, those cytokines related with the control, the regulation of the inflammatory response, they are not reaching the same amount as here in the normal women. So we have a combination of effects. On one side, we have increased amounts of the inflammatory cytokines without a compensation with the regulatory anti-inflammatory cytokines. And here you can see that despite we have a response in the inflammatory cytokines, we have enough anti-inflammatory cytokines to keep the inflammation in a very specific tone that we believe it's lower in these women and higher in this. So maybe we are, in this moment, this is only an association. We haven't studied the mechanisms, the very specific mechanisms in detail, but this is very clear that changes in the composition of the microbiota in this area of pregnant women is related with pro-inflammatory responses. And inflammation is strongly related with the development of preterm labor. So my group is composed by a clinical site and the laboratory team and the clinical team. And we really appreciate the great help that the local government is giving us to support the clinical area that we are giving these women attending the local system of health the opportunity to receive a third-level attention. Usually they don't receive any attention. They go to the hospitals only for delivery. And now they are receiving a very, very careful attention during pregnancy, and that's improving a lot of things besides preterm labor. And my group in the Institute, most of the work that I just showed was done by Berenice Palacios. And if you have any questions, you're welcome.
Thank you for this talk. I was wondering, does it mean that we could imagine that we could build or construct the vaccines against, to help preventing premature birth?
Yeah, I don't know, but maybe the use of probiotics is something that is going to happen in the next years. And but first we have to have a very specific information about the local normal composition of women, and maybe we have to group these women in different stratus or groups, yeah, in both for the normal keeping the normal composition and modifying the pathological conditions. It's not the same for every single woman. Yeah.
Okay, since you have been doing this association between the presence of certain microbiota changes and inflammatory responses, and you somehow suggest that the inflammatory responses are the actual drivers of the preterm, have you envisaged, let's say, giving anti-inflammatory therapy to prevent preterm birth?
Yeah, this is something that has been in the literature for some years, that inflammation, are an axis for explanation for preterm labor. But we don't have any single clinical trial dealing with the use of anti-inflammatory drugs for prevention or treatment of preterm labor. Maybe because we have learned during previous year that dealing with the control of inflammation in pregnant women can have other consequences on both the mother and the fetus.
So it's not that like you can, let's say, give anti-inflammatory prophylactic treatment or something to kind of unfix something that was already working.
Yeah, that's something that our main constraints are that we don't know how they're affecting the pregnancy by itself. So many people is working on that with animal models. Mm-hmm. And what we have learned until now is that they don't are the same as the human pregnancy. And the information we have available on the use of experimental models is not applicable to humans. So that's a big problem. But we are very near of that. I believe many pharmaceutical labs are working on this control of inflammation. Maybe we have to reach a very specific area, not with the systemic effect, but what's happening inside of the intrauterine environment. Yeah.
Perhaps immunotherapy against the particular cytokines?
Maybe, yeah. That's a new way. We just reported for the first time that the immune response, dealing with the specific response, the adaptive immune response, is linked with labor. So maybe we are going to modulate the role of some lymphocytes in the future. Yeah, okay. Maybe. Yeah.
Thanks.
Thank you for your wonderful talk.
Thank you.
Excellent work. I have so many questions, I don't even know where to start. But I guess I'll just ask the most pressing one in my mind. And that is, placenta is an immunoprivileged organ. And so I assume these bugs will just hide out sort of at the maternal-fetal interface. Mm-hmm. And so one question I had was, how is it possible that they can enter through circulation? I understand how it can enter through the vagina and through the birth canal, essentially. But so that's one question. How is it possible that one of your hypotheses that it goes through circulation if the placenta is an immunoprivileged organ? Yeah. And something you just answered to the last question, that it's probably the adaptive immune response, or you might look at lymphocytes. Yeah. That was also a question in my mind. What is the source of the production of these cytokines? Is it T cells, or could it be macrophages that are in there? I think that would also be important to understand, not just look at the levels of cytokines, but also to see what is the source of those cytokines, so that therapies could potentially be directed against those cell types specifically. And then the last sort of also question/comment is about ethnicity and race-specific factors. That's undoubtedly the case, and we had a large study with about 700 families where we did whole genome sequencing, and we found a lot of ethnic-specific SNPs essentially that were correlated with preterm birth, and many of them were inflammatory pathways.
Okay.
And so it's possible that not only is the microbiome ethnic-specific, but also the genetics are ethnic specific, and there is an interaction between the immune system and so it's very complex, of course. But just wondering what your thoughts are on some of those questions.
The information we have about the transplacental migration of microorganisms is coming from clinical cases, description of clinical cases in which the only explanation is that something is traveling through the placenta, but we don't have direct information of how some microorganisms can travel through the placenta and the fetus. We don't have that information, at least not in humans. We have some models, but this is not very clear. But there is something that, for example, with malaria cases in which it's very well documented that this microorganisms is reaching the fetal site. So the only explanation is that it's crossing the placenta, or maybe another vessel around the area that they are traveling to the fetus. So we don't have very specific information on that. But you have to invite me for another talk about about what cells are involved in the secretion of these cytokines. And we know that in this area, the choriodecidal area, once pregnancy is at the end of gestation, a group of immune cells are accumulating in that area, going from nothing to a lot of cells traveling to this contact area between the mother and the fetus. And they are very specific immune cells. That's our contribution. We haven't received a lot of feedback on these findings because it's very recent, but we believe that those cells, mainly T lymphocytes, a very specific type of T lymphocytes, are dealing with all the responses in this area. So maybe they are the key cells for the response, both under normal conditions and under infection conditions. And the last question is...
The last one was just a comment about ethnicity and the fact that there could be SNPs in...
Of course.
Yeah, in
we have information in the previous reports dealing with the comparison between the microbiota of women living in the same city, but with from different race, Afro-American women and Caucasian women. That's completely different. So, of course, there is a relationship between the genetic background, the response for the inflammation, and the microbiota. Yeah, of course. Yeah.
Thank you.
Just a quick question, because Ilya's questions actually start a doubt in my mind. Are those T2 lymphocytes? Yes.
They are CD4 positive and CD8 positive.
Yeah, CD4 and CD8, but those are from the T2 lineage?
Oh, yes.
Because these are the ones that are mainly present in cancer infiltrates?
Yeah.
So those are able to let's say, disrupt through membranes and kind of infect. So maybe those are the ones that are likely to diffuse through the placenta, right?
Yeah. All these lymphocytes that we are identifying in this area and in that moment are related with memory functions. Mm-hmm. They are responsible of the response to something that is known. So maybe this is related with tolerance in this area, and the rupture of that tolerance is provoking, mainly mechanistically, the labor delivery. Maybe. Okay. That's our idea.
You could
Yeah.
sequence the T cell receptor. Sorry, I'll stop.
Oh yeah, yes, of course. That's next. Yeah, once we have money for that, that's really expensive. Yeah.
Is there any other question?
I was thinking that you have a successional process in the placenta and the vagina. And I would like to know if you have analyzed this change in the species composition through time of the species, and if you have some way to understand how species are interacting.
Oh yes, we are dealing with that now. We are doing the metagenomic approach to this bacteria, and we are going to study that. All these new batch of samples, we are processing them for the shotgun metagenomics. So we are going to know every single gene that is expressed in this bacteria.
And how you can know how species interact?
Okay.
I mean, positive, negative, neutral.
Yeah, we don't know, but that's something that is going to help Enrique with.
Okay.
It's Santiago, maybe. Yeah.
No? Well, let's thank Professor Vadillo again.
Thank you very much.
And we will meet here at 3 again for the last session of the conference.